DNA replication or DNA synthesis. The purpose of DNA replication is to produce duplicate copies of DNA needed in cell division. It also duplicates the genetic information contained in each DNA. The duplicate copies will all pass to the daughter cells during mitosis of cell division. DNA replication is semi-conservative. It consists of a pair of old and new strands.
To start the initiation process of replication, the helix unwinding protein or HUP unzip the DNA double helix molecule into single-strand DNA. To avoid reannealing, helix destabilizing proteins or HDP bind to separated strands, and by using the ATP, it breaks hydrogen bonds between the nitrogen bases. Due to strong tension resulting from unwindingly process, it used gyres to release tension. Once the template are opened by HDP, short segments of RNA are synthesized by RNA polymerase and act as a primers, and it provides the 3' or OH required by the DNA polymerase III to catalyze. The leading strand opens in the direction of unzipped fork 3' to 5' direction, while lagging strand opens against unzipped fork or 5' to 3' direction.
The elongation starts when DNA polymerase III adds deoxyribonucleoside triphosphate. The complementary strand of the leading strand elongates without interruption and its direction is towards the unwinding area, while the complementary strand of the lagging strand elongates with interruption and its direction is opposite to the unwinding area. The short segment of DNA between primers is called Okazaki fragments. DNA polymerase I proofreads the DNA molecules. It removes the errors in primers and replaces it with DNA nucleotides but leaves a break or gaps. Finally, an enzyme called ligase seals the gaps left by the DNA polymerase I.
The transcription process starts when the sigma factor attaches to the double-stranded DNA to help RNA polymerase recognize the promoter sequence or the initiation point. After the RNA bind to the initiation point, the sigma factor dissociates to participate for another round of initiation of transcription in other sites. The RNA polymerase enzyme splits the double-stranded DNA molecule and produces a segment of complementary RNA strand to the anti-coding strand of DNA using uracil instead of thiamine. Since attraction between DNA strands is stronger than DNA and RNA strands, the double-stranded DNA molecule reanneal and displaces the newly synthesized RNA strand. Terminators called raw factor attaches to the termination site, signaling the RNA transcript is complete and causes the transcript to be released from the DNA polymerase, producing the newly synthesized mRNA strand.
Before the translation initiate, there must an activation of amino acid tRNA complex by adding tRNA to an amino acid formylated methionine with the help of amino acid synthetase. And then initiation factor 1 carries amino acid tRNA complex attached to small ribosomal subunit or the 30S, and initiation factor 3 carries mRNA to the starting codon AUG. The initiation factor 2 carries large ribosomal subunit or the 50S, then attached to small ribosomal subunit to form 70S. This 70S is divided into P site and E site. P site is the exit site while E site is the entrance site, and elongation process undergo by adding another amino acid tRNA complex to other amino acid tRNA complex. When it reaches the release factor 1 and 2, ribosomal subunit dissociates to the amino acid tRNA complex.
The purpose of translation process is to synthesize protein.
Structural proteins have been discovered in the human body, but most abundant by far is collagen, which makes up about 6% of total body weight. Collagen makes up 30% of bone tissues and comprises large amount of tendons, ligaments, cartilage, skin, and muscle. Although proteins are found in greatest amounts in connective tissues such as bones, their most extraordinary function as enzymes. Enzymes are proteins that conduct specific chemical reactions. An enzyme job is to provide a site for a chemical reaction and lower the amount of energy and the time it takes for the chemical reaction to happen.